Right, good afternoon. My name is Peter van der Spek. I'm working at the Erasmus Medical Center. It's one of the largest medical centers in the Netherlands. We basically have under one roof a cancer hospital, a general hospital, a children's hospital and a harbor hospital. What I'm going to show you is what we are doing in the field of translational medicine in my talk, and later on we can then discuss several items that come along here. So basically the Department of Bioinformatics is integrating data from patients, drug treatments, expression profiles, functions of genes or proteins that scientists and clinicians try to study. And by integrating the data in an efficient manner, you reduce it, come towards information, which leads to knowledge. And obviously we try to understand which changes in our DNA, for instance, can lead to the cause of cancer or are important in congenital malformations. So what you see here on this slide is one of the activities that we have started to deploy here in the Bay Area roughly a year ago. We are aggressively sequencing human genomes at the DNA level with a company called Complete Genomics, and roughly we generate per sample 1.5 terabyte on human variants in the human genome per sample. So by stacking these alongside each other, we've built, in collaboration with the folks from Oracle, a database to query along the huge data sets the variants which occur in cancer samples, in normal samples, and in samples derived from congenital malformations. And what we see is this human variome database in the middle. We are loading that with information from clinical traits, which are indicated on the right-hand side. We load it with functional expression-based data that we have from our Affymetrix array platform that we use to study transcriptomics in models, in particular in human systems. And then obviously we want to rank the variants that we pick up and the candidates which are most likely to be involved in the disease. And then you start to test your hypothesis using pathway tools, for instance, from Ingenuity, and you go to animal models or to other patients to validate your observations. Now what we realized is that building a database is very important because you need that for your support of your research, for understanding in future and running your diagnostics more efficiently, but also to collect and analyze the data which is derived from your clinical trials. Now, as pharmaceutical companies deliver their data frozen in Oracle onto the FDA, a hospital needs to store its data from the patients that they have sequenced even 5 years after the patient has died, and your electronic healthcare archives are also organized by databases which are coming from Oracle. So we saw a very interesting niche where actually this database could be a very central point which basically allows us to integrate data but also to facilitate research in a very basic environment, also facilitate epidemiological studies, and obviously host data derived from clinical trials. So what you see here is all kind of views that we generate on data. I'm not going to specify specific analysis here, but these are the typical visuals that scientists are asking for. And obviously, referring to the talks where we have the public-private partnerships, what we see here is the model build, bundle, benefit, where the build exploratory research is funded usually through the Ministry of Education. The middle part is basically indicating the translational bit by bringing it into the hospital and applying it towards patients in a proof of concept funded through the Ministry of Healthcare. And then on the right-hand side, a startup company is formed if the proof of concept seems to work. You want to bring the technology towards the patient, and obviously this is not something what a hospital should try to do. Hospitals are there to treat patients and companies are there to sell products. So this is definitely a clear effort which is funded through the Ministry of Economical Affairs, and one of the companies that spun out of the Department of Hematology and my Department of Bioinformatics is called the company Skyline. Well, that's a very nice name for the city of Rotterdam, but also here, well, I have to say this is a very impressive skyline that you have here. So why did we think of that term skyline? I always explain to the students, it's like basically stratifying patients into groups. So if we make a snapshot of a city, of a skyline, 30,000 buildings. A tall building has a lot of RNA, a small building is a little bit of RNA, and I'm making pictures of all my tumors that I'm comparing. And then I'm going to sort these pictures onto piles. And these piles basically reflect what's going on. If there's a block in the blood cell differentiation, they have a very similar skyline, these patients. So by binning them into groups, we can target them for targeted treatment. So by selecting them and stratifying them on their own characteristic skyline, that's a method which works very well in our hands. So we have set up very strong biobanks, which was already referred to in the previous talks. So the biobank with the leukemia samples, we apply omics technologies, link with the electronic healthcare archives. Some of the patients are enrolled in clinical trials, and obviously we have a lot of researchers who do all kinds of investigations with these samples. The overall aim is to come towards better diagnosis, earlier intervention, more effective therapy, improved health outcome, and eventually we also are thinking about increased cost effectiveness, of course. So what's very important to realize is that 4% of the healthcare costs are spent on diagnostics. 72% of the decisions is based on this diagnostics. So by investing 1% more, the impact on the back-end side is substantial. So just to give you an idea, in our children's hospital, 1 in 100 children that's born gets a congenital malformation. 40% of the activities in our children's hospital are activities that link to these 1 in 100 individuals. So that's a very, very significant number, which points to, if you more effectively diagnose what's going on in these children and not have to do 20 different tests, but immediately sequence the genomes and stack them alongside and nail where are the most relevant variants, that gives way more insight, and also obviously you are more accurate and quicker in getting an idea about what's going on. 1 in 3 or 1 in 4 currently gets cancer. Our cancer hospital sees obviously these patients, and that's going to move to 1 in 3 with an aging population. And in the bottom you see all the 8 university medical centers, and what we currently are doing is set up with all these centers collaborations around this next generation sequencing technology. We are basically showing what we have done with our 100 genomes over the last year and how we are going to ramp up with an additional 250 samples in the coming year, because we see that the uptake and the implementation of the technology definitely makes sense, and for a medical center to write off these fast developing equipment is basically not cost effective. So we are very interested in the business model that Complete Genomics handles to actually write off equipment on their side, and based on the economy of scale actually provides very interesting ways of working with medical centers and deliver standardized technology, which is obviously very important also if you think in terms of doing multi-center clinical trials. So what you see here is an example of the Skyline work. We keep studying the same samples every time when there's a new technology. We keep throwing new technologies onto the same patients, from which we have built up a very, very clear picture at the molecular level in a historical manner. And on the left-hand side, you see the molecular expression-based perspective, where we have divided the patients into groups. These red blocks indicate characteristic skylines, and on the right-hand side, on these diagonals, you can actually see the morphology indicated in the different colors in these blocks. You actually see the number of white blood cells indicated in a proportional manner. Mutations in a prognostic gene, they don't distribute randomly, they go for very specific skylines. You can actually see the overall survival and the event survival, free survival in the top part, which is for certain groups very much better than in other groups. So by stratifying based on these skylines definitely makes sense, but we also have subgroups for which we don't know yet what are the driver mutations, and that's why sequencing with Complete Genomics, the full genomes, opens up for us new candidate mutations which are very important to pursue in this kind of cohorts. So what we actually have implemented very efficiently with the leukemias, which are indicated on the top left, at the DNA level and at the RNA level, we started applying the same technologies also in the area of lung cancer, colon cancer, brain tumors, breast cancer, prostate cancer, and liver cancer. These studies initially all have been done with RNA and with DNA expression arrays, and now the areas where we want to have more understanding and more detailed information, we are going to apply sequencing technology, and from the sequencing technology we find new mutations which we eventually can array again, and then actually both technologies can occur very well alongside of each other, we think. And what we see here is the different sizes of the groups, which have their own characteristic skyline, and obviously from a pharmaceutical perspective and a commercial perspective, you rather want to work on the largest group first. Well, that's not how biology usually turns out in science. You do unexpected findings, and actually in this rather small group which I'm pointing at the moment, that group actually expresses a target for which a drug was developed by Roche, which is called Herceptin, and actually only that group expresses the target for Herceptin, so only that group potentially can benefit from a combination therapy. So also the way of thinking by first stratifying patients into group and then think what are the most efficient therapeutic interventions that can be applied to these patients is going to change the medical landscape substantially. So obviously we do this for the patients, and actually what turns out, this patient actually did very well after his bone marrow transplant and won a golden medal at the Olympics. So what we see here on the left is that the molecular profiling has resulted in biomarkers. Obviously we want to bring these biomarkers towards the imaging field, what Peter Luijten already referred to. And well, obviously what we see on this picture, we've built a cinema in our department where you can project MRI, CT, ultrasound images in 3D and walk in 3D through these data sets and explore them. And what we also can do is actually paint in whole mount in situs protein expression or expression of a RNA molecule, and actually see where a gene at a particular moment in time has its particular role, or whether it's absent or whether it actually is a important gene which explains a congenital malformation, etc., etc. So linking the imaging modalities towards the molecular work is also something what's on our agenda. I'm going to try to explain you some final activities that we do in the area of development of the head and the face, where we have an interest in. We describe processes which are important in the development of the brain. If the genes involved in these different processes, if they accumulate a mutation, it causes malformations, and obviously they occur at different moments in time. So by monitoring over time in a longitudinal manner, you can get a much better understanding, but that requires infrastructure to store and capture that information. So here we see medical images, definitely a normal brain on the left top part, a brain with a too thick cortex, a brain without gyri, a brain with polymicrogyria, so too many and too small gyri, a band heterotopia, so there's an additional band visible here, and over here basically neuronal migration didn't occur, so you see a lot of gray matter here along the ventricles, which was supposed to migrate out to the side of the cortex. So it's not only genetics, it's also lifestyle and environment. So basically a polymicrogyria can occur due to a genetic mutation. It can occur due to excessive alcohol intake during pregnancy or cocaine abuse, and obviously a viral infection with a cytomegalovirus virus from external also can cause a polymicrogyria. So there are three areas from which the polymicrogyria, for instance, can occur. So by screening every 1 in 100 doesn't mean that every 1 in 100 every 1 in 100 has a genetic malformation. So you screen actually more, and not everything turns out to be genetic. Now what we see is the impact of information technology. Information technology makes things possible to what I'm showing here is only possible due to the rapid progress in the IT sector, which is mainly driven here in this Bay Area. So what we are doing, we build clever databases, but also we want to mine these databases in a very easy manner, make use of modern technology like iPads, and obviously define and report back as efficient as possible to the clinician so that he can show in the polyclinic very directly to the patient what's going on. So different models require different queries on the database. The reporting goes back on the iPad, and that's how it gets distributed back. Now, these are the papers that are coming up in 2 days. So in July this paper will come out. It's the first mutations that we start publishing now from our studies that we have done over the last year with complete genomics. We very efficiently sequenced father, mother, child pairs where we find very novel mutations, and this is actually a mutation which is important in a gene which causes craniosynostosis. Now, this is my favorite picture to outline what's going on with genome sequencing. It's very IT driven, and obviously half of your genome you get from your father, that's the four-wheel, and the other half comes from your mother. And as you can see, these seed variation length is 90% genetically determined. That's what our forensic experts have figured out in our institute. And actually, obviously, by looking at those variants in your genome, what we are doing with the forensics division is finding external traits which you can link back to variants in the human genome, because these are the ones which you can use to make new profiles of individuals who have been relevant or are relevant at a murder scene. So we can delineate from a drop of blood external features to indicate this is an individual which is a Western individual, blue eyes, length roughly this and this length. So by making profiles based on the DNA variation, this is a very important topic, in particular in our forensic activities. Also, training the clinicians becomes very important if you want to apply these new technologies. So we are making education decks where we apply SNP technology, so array technology in conjunction with next-gen sequencing technology, and building very specific education sets. We can start to train clinicians which are relevant to interpret the data if you start to apply these new technologies more and more in the near future. Some of the things you cannot detect with an array, but you can detect them obviously with sequencing. So these technologies occur very well alongside each other. So here we see an example with array technology of a particular deletion. Here we have the medical images, and obviously in the animal model then we see very specific validation, staining of the particular region, which is important during development of the brain. This is in embryonal stage E11 in mice, but actually if this expression is not occurring at that particular moment in time, it causes a malformation of the cortex of the brain in this particular child again. Now, what we have observed with our expression data is very fascinating. Actually, the growth of the head, and if you measure the circumference, we see in dorsal lateral cortex of the developing brain. This is from children, human material, from children of 0.1 year, 0.2 year, 0.3 year, all the way up to individuals of 80 years old. Same region of the brain, very specific staining until the age 3. Now, the genes which are basically boxed here, these genes are very important in the development spurt, which you actually see in these growth curves, here for females and here for males on the left hand side. So these are very important growth related transcripts. Now, after age 3, autism becomes apparent. So the genes which are in this boxed area are very important to monitor more in detail in autism. So having expression data, overlay that with next-gen data where you very rapidly, all the 3 billion base pairs are monitoring for variants in particular genes, then you will find the enrichment in actually the mutations that occur in these transcripts that we have picked up with the FE platform. So here we have an atlas of an adult brain. So 53 regions have been biopsied, all from independent healthy donors which died because of a car accident or a cardiovascular problem, not a CNS related disorder. And then we have the DTI data, the fiber tracking data, on seeing which areas project into each other, and then actually we can see which transcripts are very important in which area. So what we see here is that we sequence full genomes. The reason why we do that is these orange, yellow indicated areas. So why is that? The enhancer elements, they're the non-coding bits, but that's the transcription factor binding sites and obviously enhancers for transcription complexes like you see here. And actually in blue you see staining of enhancer regions related to the developmental areas during embryonal development. And we studied that for the forebrain in detail, and here we see all the loci that we are monitoring very closely for variants that occur also in our craniosynostosis families that we have collected. And obviously in the 3 areas of the development of the head and the face, we monitor this again in time series with expression arrays in order to have functional validation along with the variants that we have picked up with the DNA sequencing. So we think that basically combining molecular with images gives new opportunities for disease management. Expression-based classification of biological subgroups is very important for targeted treatments. Identifying novel DNA variants obviously is very relevant also for prescribing novel therapies. The development of companion diagnostics, I have shown you some examples of what we are doing in the field of leukemia in order to actually identify which patients are eligible for a bone marrow transplant and which ones are basically doing well with a vitamin A derivative which is called ATRA. So that's basically a drug that you give to push the bone marrow differentiation, and which works very well, for instance, with the good prognosis group that I've shown you with the good overall survival and event-free survival. And obviously assessment and management of risk is very important for the patient. So last but not least, obviously we talk about patients. So what we see in this last slide, a lot of institutes, public and private centers with whom we collaborate in our scientific endeavors where we try to apply novel molecular technologies that are coming usually from the Bay Area. So thanks for your attention, and I would like to give the floor back. Thank you.